the ICGC TCGA Dream Somatic Mutation Calling Challenge. We've got 3 speakers. First, I'll give an overview to the challenges themselves and to the general problem of sequencing data quality in cancer genomics. After that, Dr. Anna Lee will talk about the results of the structural variant benchmarking. And then lastly, Dr. Ken Chen will talk about NovoBreak, the algorithm that's been winning some of the structural variant benchmarking components of this. So the key thing that we're going to talk about is the variability in analysis protocols and the consequences that has for our interpretation of cancer genomes. Then we'll talk about SMC-DNA and how we've used that approach of crowdsourced challenge-based benchmarking to come up with a better understanding of the analysis variability. And then I'll talk about the ongoing and future benchmarking efforts. So as we all know, the basic protocol that we follow is to have some sort of large genomic data set generated. Whatever this is, it could be ChIP-seq, it could be RNA-seq, whatever type of data it is, we're looking to try to understand what relationship it has to the genomics of a cell, or in terms of tumors, to patient survival. And in some way, therefore, our ultimate consumers are molecular biologists and clinicians who want to interpret that data, and they treat the genomics and the computational biology as a bit of a black box. The black box of data analysis to which we put in precious patient samples and hopefully come up with an answer that is going to help us yield a clinically relevant conclusion. The problem, as we know very well, is that if we take different types of black boxes, different ways of analyzing the same data set, we don't always come to the same conclusion. And that's been shown over and over in a variety of different contexts, and I'll briefly review a couple. For example, if we were to take a look at mRNA-based biomarkers to predict the outcome of patients with lung cancer, and we look at 24 different ways of pre-processing the data set, we can see very poor agreement across the different pre-processing methods. Here, each of the columns is one of 442 patients used for validation. Each of the rows is a different way of pre-processing the data, and you can see that only a third of patients show a consistent response, a consistent prediction, independent of the way that we pre-process the data. And of course, you could say this is mRNA data in one tumor type, and microarrays are particularly prone to pre-processing and batch effects, so maybe this isn't general. And if we were to look at other tumor types like breast cancer, we can see the exact same profile. And here, different columns are different pre-processing methods, and you can see the number of genes that are being altered. And in fact, in breast cancer, the problem is even more severe. 74% of genes show an association with patient survival in at least one of the different pre-processing methods. So if we were being intellectually dishonest and somebody says, is this gene associated with breast cancer, with a 74% chance, we can say, yes, it is in one of the different data sets. But by contrast, not a single gene was consistently associated with patient survival in at least 21 of the 24 different pre-processing methods. So you see this huge variability. And this, of course, holds true also in the area of next-generation sequencing. You're seeing on the left results from one of the TCGA unified genome calls where 3 major sequencing centers, Baylor, the Broad, and UCSC, made calls on a series of patients from the renal project. And as you can see, there's a core of mutations, about 12,000, that are found by all of the different sequencing centers. There are also very large numbers that are only found by one sequencing center or another. And I want to emphasize, although the sequencing was done at a single center, these are different analysis pipelines, not different reads from different parts of the tumor or anything. It's just a simply reanalysis of the same FASTQ files. Similarly, in work done in my lab on structural variant calling, you see the exact same problem, where different algorithms yield very diverse results. So this has major consequences for our ability to use next-generation sequencing robustly. And so to try to resolve that issue, we launched the ICGC TCGA Dream Somatic Mutation Calling Challenge. I'm just going to call it SMC, or the challenge to make it simple. And the idea was that to try to resolve this problem, we would set a series of standard data sets that we would make available to groups around the world and allow them to start doing an analysis, and we would act as an honest broker to assess the accuracy of those different analyses done on that. The challenge itself started in 2013 in December and ran through almost until the end of 2014. It has two major components. One is an in silico tumor component, where we developed BAM Surgeon, a tool to be able to robustly simulate tumor genomes starting off with a normal genome sequence. And we released 5 tumor normal pairs from that. And those were made to be of increasing difficulty, starting off with a fairly easy tumor and then building up to a very complex with significant subclonal heterogeneity tumor. There were also 10 tumor normal pairs that were real sequence tumors, 5 pancreatic, 5 prostate, and for these we made lane level FASTQs as well as BAMs available, and we tried to make this available in as many formats with different aligners so that groups could try to use their pipelines in whatever way they would like to test them. The data itself is still easily available. You can register for the challenge even today and be able to submit to the last of the 5 in silico tumors. There's a templated ICGC DACO agreement which just allows you to get access to the confidential sequence data, confidential meaning private human information, pretty readily, and that takes about a day to do the application and get the approval. Anai Biosystems also provided free download access through the GeneTorrent client, and Google provided free computing and access to the data in the cloud. And so far, we've had about 400 groups around the world participate in this, making over 3,200 individual predictions of mutations across 14 genomes. I mentioned the last genome is still in progress. And this has led to a lot of really interesting things. Most importantly, and gratifyingly, people have continued to use the leaderboards and the tumors as a gold standard to assess new methods. And so even after the challenge is being done, this is becoming a sort of living benchmark. And it's our hope that journals will start to expect people to look at the results on these tumors as part of standard submission of new algorithms. There's also been some really interesting discussions about the scoring of structural variants, which Anna will talk about afterwards, as well as on improving our ability to accurately simulate cancer genomes. And so I'll show you some of those really interesting results that we've had to date. What you're seeing here is a precision recall curve for 2 tumors. The first tumor on the left is the simplest tumor. It's 100% cellular with no subclonal populations. It's a very easy tumor, and you can see that groups do very well. Although there's some sort of a PR relationship there, in the inset you can see that the top groups are getting something like 97 or 90%, 98% recall with an F-score of about 0.96. Those are really robust numbers, although it's worth noting, even there groups are missing 2 or 3% of mutations, even on a tumor that is as easy as we could make it. Now when we introduce contaminating normal cells and remove to an 80% cellular tumor, you can see the recall rate shift down by about 5%, increasing the false negative rate, or representing an increased false negative rate from different groups. We were worried that groups might be overfitting, that we had released a small amount of sequencing information, or sorry, of validation information to allow people to access the leaderboard. And of course, having the ability to resubmit to the leaderboard could lead to overtraining. And in all of the tumors, we saw no evidence of groups overfitting. In fact, what was really interesting is that over time, groups were able to converge on a more correctly fit answer by retuning their algorithms and reparameterizing. There's a number of interesting features of the errors that different groups were making. In particular, it turned out that the coding regions had substantively lower error rates, almost 8 to 9% lower error or higher accuracy than intergenic regions, which may reflect different amounts of repeats in different regions of the genome, or may simply reflect tuning of algorithms on the coding regions for groups that have been developing them. But even in the coding regions, there are recurrent errors. There are regions of the genome that are only being called within the coding, individual point mutations that are only being called by 2 or 3 groups. And what's really striking is that of the top 20 teams, only 3 called the coding region perfectly accurately. So even in this easier portion of the genome that you might think is the most functionally relevant for point mutations, only a small number of groups were able to exactly identify point mutations. We also looked to see if one could develop ensemble callers, if we could use multiple pipelines together. And you can see here that as we added individual pipelines, moving across increasing ranks, the ensemble caller in gray continued to be very stable and to outperform individual callers. And this may provide a really interesting avenue for improving our pipelines, rather than using a single tool, using a panel of 4 or 5, and not worrying as much about the parameterization of that tool for an individual tumor, but thinking about how multiple algorithms can come up with a consensus call. But despite that, parameterization turned out to be critical. What you're seeing here are individual submissions by individual groups and their performance. So in this white bar at the top, or sorry, on the left, you're seeing the best group, the one that won this particular tumor. And you can see that they made 2 submissions, one that was quite good and one that improved significantly. You can also see that many other groups made multiple submissions and were able to significantly improve their performance. For example, consider the third team. They started off with an accuracy of about 75%, or an F-score of 75%, and then were able to increase that almost to 92% simply by optimizing the parameters. And many groups were able to do this quite consistently. This may indicate a flaw in the way we think of genomic experiments. We think we generate a data set, then we analyze it with the best available tools. And maybe a more appropriate methodology would be we analyze a data set, come up with an initial set of predictions, validate a small number of those, and then use those to refine and reparameterize our algorithm. And we were able to see that some groups were very consistently good at parameterizing their algorithms and improving it over time. And in fact, it's probably an underexplored area, the auto-parameterization and tuning of algorithms for individual tumors, something that we should all probably think a lot more about how this could be automated and thought through. So we may have a lot of opportunities here between ensemble calling and improvements in parameterization without even developing new algorithms to improve our ability to robustly call variants in cancer genomes. There's a lot of other features to the error profiles that are quite interesting. For example, if we take a look at the false positives and we take a look at their trinucleotide profile, that is the base 5' and the base 3' to a false positive, they have a very specific and very biased false positive profile. And in fact, this profile is the one that is associated with deamination. It's identical to the profile of human aging that is widely seen in human tumors. And so you can see now that the variant callers can be making errors that would confound certain types of analyses. And you may imagine that you would want to use different algorithms for different types of questions based on the error profiles of the algorithms and the characteristics of the question that you want to ask. So the work that I've just described is now in press at Nature Methods. Actually, I think it just came out last week. And you'll hear in a moment about the structural variant analysis from Anna. We're also going to have the experimental validation studies of the real human tumors ready for release soon. And those involve deep resequencing of several thousand variants, both single nucleotide and structural variant, for each of those tumors, which we think will make a robust and very useful gold standard for the community. Given the success of this, we're also looking forward to other types of challenges. And in particular, we're looking at 2 things. The most imminent to launching is a SMC Het challenge, which is looking at subclonal reconstruction. And a little further off, late this year or early next year, will be one based on fusion proteins from RNA sequencing data. I'll talk quickly about the heterogeneity challenge. So you can imagine that the same amount of variability that you see across pipelines occurs in every area of bioinformatics. And as part of the TCGA prostate project, which is now submitted to Cell, we were looking at normal cell contamination, and we really means a group of people up here. And we looked to see if we could take a look at the estimation of infiltrating normal cells using different types of data. Some types of data that came from mRNA abundance, some that came from microRNAs, many that came from DNA sequencing data, and we thought we would find robust conclusions where the same estimates of tumor cellularity would be inferred by different algorithms. And of course, that wasn't the case. Most of the algorithms agreed with each other about 50% of the time. And so we started to think about the many reasons why this could be. And as a result, we launched the ICGC TCGA Tumor Heterogeneity and Evolution Challenge. And this is something that is going to open for actual submissions in August. And the exact date, we don't know yet. I say August 2015. I think it'll be about the middle of the month. The idea here is that there's going to be two separate questions asked. The first is a single sample question where we give groups a single set of variant calls from a single tumor genome. These will be simulated tumors using the genome in a bottle reference as a basis. And groups will be asked to infer things like the number of subpopulations, their cellularity, and characteristics like that. What's really unique about this is that it'll be a cloud-only challenge. That is, people will be submitting Dockerized instances ready to be run in Galaxy workflows, and that'll be the main method or the only method for people submitting. So as a secondary result, there will be a stable of algorithms with a consistent API, consistent interface, consistent scoring standard as a community-generated standard for all analyses of subclonal reconstruction afterwards. There will similarly and sequentially be a multi-sample challenge where we look at multiple regions from a single tumor. The simulated tumors will be built using BAM Surgeon, but BAM Surgeon has been adapted to be able to handle phased reads and phased mutations. And we've done a series of validation studies to demonstrate that this is a robust way of working with it and it yields similar results to real tumors. And so I'll skip mentioning the simulating, but say that at the end of the challenge, we will be making the BAMs as well as all of the variant calls from multiple algorithms available, as well as the series of Galaxy and Docker instances as a large-scale resource. We have a tentative design in terms of varying cellularity, varying the subclonal structure and characteristics like that, and that as well will become a final product of the challenge. And so I'll close off by saying that the area of benchmarking cancer genomics algorithms is critical. It's something that we need to be able to do better. And there's a lot of value in the crowdsourced benchmarking approach. And I'd encourage everybody to get engaged with this if they have algorithms. It's relatively easy to do, and we try to make the interfaces relatively clean. And Google and other companies are donating significant amounts of cloud computing time to allow you to be able to run this readily without any challenges. These are the people that were particularly involved in the SMC-DNA challenge, and in particular, Katie Houlihan and Adam Ewing did much of the work that I showed on screen, and Anna will talk in a moment about the structural variations, and these are the people engaged in the SMC-Het challenge. Thank you very much. So we have a minute or two for questions now before we move on to the next talk, if there are any.
I could probably ask you this at work, but what about Dockerizing the existing SNV challenge?
So it's a very bright light, so I actually couldn't recognize who it was until I saw heard the voice. So Michelle, it's a great question. We are not asking for Dockerized versions of existing methods because the challenge is complete and we thought that would be unfair. But what we are doing is preparing to launch a second iteration of this challenge with longer read sequencing, with updated sequencing protocols. So this version, for example, used a PCR-based protocol for the sequencing libraries, and that's rapidly falling out of favor as improved genomic ability to using no PCR libraries is coming into play. So it's likely that the next challenge will also be a cloud-only one that will try to account for that. All right. Let's move to Anna's talk. So Anna will come and speak about structural variant analysis in the SMC-DNA data set.